All right, let's get right into it. We're going to unlock the code of our blood types, what really makes them different, and why it matters so much.
So, we'll kick things off with this really critical question. We all know that giving someone the wrong blood type can be catastrophic, right? But why? The reason is actually a fascinating story, all about microscopic ID tags and a kind of biological security system that's working inside every single one of us.
Okay, so what are the actual rules of the game here? Let's lay out the basics of who can safely give blood to whom.
Now, this chart shows the fundamental rules. You've probably heard that type O folks are the universal donors. They can give to anyone. And type AB, they're the universal recipients. But look, these are just the rules. The really cool part is figuring out the why behind them. What is actually happening inside our veins that makes this so incredibly important?
To really get it, we need to meet the two main characters in this story. The best way to think about them is as a system of ID tags and security guards that are just floating around in your bloodstream all the time. First up, we have the ID tags. They're officially called antigens, and you can picture them as tiny little flags sticking off the surface of every single red blood cell you have. They're constantly announcing their identity, like, hey, I'm a type A cell, or over here, I'm type B.
And next, meet the security guards. These are your antibodies. Now, they patrol the liquid part of your blood, the plasma, and they have one job and one job only, to hunt down and attack any red blood cells that have a foreign ID tag, one that doesn't belong.
So this is the really crucial thing to remember. Your antigens, the ID tags, are literally attached to your red blood cells, but your antibodies, the security guards, are free-floating in the plasma, always on patrol, always looking for invaders.
And this picture just lays it out perfectly. If you've got type A blood, your cells have A ID tags. So to protect you, your body makes anti-B security guards. If you have type B blood, it's the exact opposite. B tags on the cells and anti-A guards in the plasma. Now, type AB has both A and B tags, so it doesn't make any security guards. It would attack itself. And type O, it has no A or B tags on its cells, so it has both anti-A and anti-B guards on patrol. It's a perfect system. You never make security guards that would attack your own cells.
So, what happens when we break the rules? What goes down when those security guards finally find an intruder?
When the antibodies attack those foreign antigens, they trigger a reaction called agglutination. And this isn't some small problem. We're talking about a massive, dangerous clumping of red blood cells. It can clog up your blood vessels and can lead to a really catastrophic immune response.
Okay, let's just run through a quick scenario. Let's see this disaster unfold. Imagine a patient with type A blood needs a transfusion right now.
The absolute second that wrong blood, that type B blood, enters their body, the patient's own anti-B security guards sound the alarm. They just swarm the donated cells, grabbing onto those foreign B antigens and binding them all together into these huge clumps. That is agglutination, and that is why blood compatibility is quite literally a matter of life and death.
So we've been talking about these ID tags, these antigens, like there's some complex piece of machinery. But what are they really? I mean, what are they made of? Let's zoom way in, down to the molecular level, and find out.
And here is maybe the most mind-blowing part of this whole story. This entire system, the rules, the life or death consequences, it all comes down to an unbelievably tiny difference. It's just the presence or absence of a single, simple sugar molecule. That's it.
It all starts with the foundation, a kind of precursor structure called the H antigen. Think of this like a blank ID tag. It's on the surface of pretty much every red blood cell. It's the base that your specific blood type gets built on.
And here it is, the big reveal. Your genes basically tell an enzyme what to do. If that enzyme attaches a sugar called N-acetylgalactosamine to that H antigen base, then congratulations, you have type A blood. But if a different enzyme attaches a different sugar, one called D-galactose, you get type B blood. The whole thing is decided by one tiny sugar. It's incredible.
So let's put it all together. If you're type O, your body just leaves the basic H antigen as is. It doesn't add that final sugar. If you're type A or type B, you add your specific sugar. And if you're type AB, well, your body has enzymes that do both jobs. So some of your H antigens get turned into A and others get turned into B. Okay, let's zoom back out. Knowing the elegant science is one thing, But how do we actually use this in a hospital, when the clock is ticking and we need to save a life?
The test is actually beautiful in its simplicity. We take a little bit of a patient's blood, and we basically provoke that clumping reaction on purpose, in a safe, controlled way. We just mix one drop of blood with a serum full of anti-A antibodies and another drop with a serum full of anti-B antibodies. And the results are completely visual. You can see the science happen right before your eyes. All you have to do is look for where the blood clumps up.
The logic here is just rock solid. If your blood clumps when you mix it with the anti-A serum, well, it must have A antigens on its cells, right? So you're type A. If it clumps in both serums, you must have both A and B antigens. That makes you type AB. And what if it doesn't clump at all? That means your cells don't have A or B antigens. You're type O. Simple as that.
And this entire elegant system was first figured out way back in 1901 by a scientist named Carl Landsteiner. He did a series of really simple but totally brilliant experiments. He just mixed blood cells and plasma from people in his lab and carefully watched for these exact same clumping patterns.
And that discovery, made more than 100 years ago, is still the absolute cornerstone of transfusion medicine. Making sure the donor's ID tags are compatible with the patient's security guards, that is the essential first step that makes so much of modern medicine even possible.
Which brings us to a really fascinating final thought. We've learned that this huge life-or-death difference between blood types is just one tiny sugar molecule. So what could that mean for the future? Could scientists find a way to just snip off that final sugar? Could they create an enzyme that turns any blood type into the universal type O?